Good day, these are the sample questions for the 7th topic for aquaculture. The fish genetics. Number 1. This implies that individuals of the same species may differ from one another, and yet they may have characteristics common to all of them. A. Genetics. B. Origin. C. Ancestry. D. Genes. Answer: A. Genetics, because it is the branch of biology which attempts to describe similarities and differences among organisms or variation and to explain the way in which these characteristics are transmitted through successive generations or heredity. Number 2. This is the genetic unit that contains the blueprint or biological code for the production of phenotype. A. Allele. B. Chromosomes. C. Origin. D. Genes. Answer: D. Genes. It is a linear array of very specific subunits or the basic unit of inheritance and is but a small segment of a much larger molecule called deoxyribonucleic acid or DNA. Number 3. In the three main areas of genetics, this refers to the principles of transmission of genetic material from one generation to the next. A. Quantitative genetics. B. Population genetics. C. Mendelian genetics. D. Genetics. Answer: C. Mendelian genetics, because it refers to the principles of transmission of genetic material, while the quantitative is relatively simple statistical concepts that is hypothesized to contribute to the expression of traits. And population genes is the study of Mendelian genetics in population of plants and animals. Number 4. This law of Mendel states that genes exist in pairs in the cells of the individual, and the members of the pair separate during gamete formation so that any sex cell contains and transmit only one member of the pair. A. Law of segregation. B. Law of independent assortment. C. Mendel's second law. D. Mendel's third law. Answer: A. Law of segregation. It is a diploid individual possesses a pair of alleles for any particular trait and each parent passes one of these randomly to its offspring. When gametes are formed, each allele of one parent segregates randomly into the gametes, such that half of the parent's gametes carry each allele. Number 5. This refers to all offspring produced through self-fertilization that are essentially identical to the parent plant. A. Breeding. B. True breeding. C. Origin. D. Cross-fertilization. Answer: B. True breeding, breeding wherein the parents would produce offspring that would carry the same phenotype. This means that the parents are homozygous for every trait. Number 6. In mode of gene actions, when the dominant allele expresses itself more strongly than the recessive allele, but not strong enough to make the heterozygous phenotype identical to the homozygous dominant phenotype. A. Dominance. B. Complete dominance. C. Additive gene action. D. Incomplete dominance. Answer: D. Incomplete dominance. When the mode of gene action is complete dominance, there are only two phenotypes because the presence of the dominant allele masks the expression of the recessive allele in the heterozygous genotype, but in incomplete there are three phenotypes and three genotypes, a separate phenotype for each genotype. Number 7. It refers to the exchange of segments of DNA of homologous chromosomes. A. Crossing over. B. Tetrads. C. Reduction division. D. Equational division. Answer: A. Crossing over. A process of reshuffles the genes and creates new combinations of genes in the gametes. Crossing over occurs when chromosomal homologues exchange information during metaphase of meiosis. Number 8. It is a term used to describe a variety of manipulations of biological systems for human benefit. A. Biology. B. Biotechnology. C. Genetic. D. Biological system. Answer: B. Biotechnology is the manipulation of the molecular basis of inheritance by methods collectively called recombinant DNA technology. It can be used to describe the application of new genetic technologies to improve performance which would not be possible using conventional selective breeding techniques. Number 9. It is a method of producing virtually unlimited numbers of copies of a specific piece of DNA starting with as little as one copy of the desired DNA. A. Enzymes. B. Genotype. C. Selection. D. Polymerase chain reaction or PCR. Answer: D. Polymerase chain reaction or PCR. It is a chemical process used by molecular biologists to amplify DNA fragments. A single or a few copies of DNA can be reproduced into millions or billions of copies using this reaction. And by amplifying that DNA, we can analyze that DNA molecule in greater depth in the laboratory. Number 10. The following are advantages of fish for biotech applications except 1. A. Fish can produce large numbers of eggs. B. They can be induced to spawn more than once a year. C. Gametes are easy to handle. D. Generate economic benefits, including improved plants and animals for agriculture and efficient production of valuable biological molecules. Answer: D. Generate economic benefits, including improved plants and animals for agriculture and efficient production of valuable biological molecules, because this statement belongs to the goals of biotech application. Number 11. The process of reducing the number of individual with a population by removing the inferior one. A. Culling. B. Elimination. C. Grading. D. Sorting. Answer: A. Culling. It is the process of segregating organisms from a group according to desired or undesired characteristics. Number 12. A mechanism where the sex is determined by prevailing temperature during the development of sex organ. A. Chromosomal sex determination. B. Autosomal sex determination. C. Environmental sex determination. D. None of the above. Answer: Environmental sex determination. Temperature-dependent sex determination or TSD, is a type of environmental sex determination in which the temperatures experienced during embryonic/larval development determine the sex of the offspring. It is only observed in reptiles and teleost fish. Number 13. The production of offspring by a female with no genetic contribution from male. A. Parthenogenesis. B. Gynogenesis. C. Hermaphroditic. D. None of the above. Answer: A. Parthenogenesis. Parthenogenesis can be defined as the production of an embryo from a female gamete without any genetic contribution from a male gamete, with or without the eventual development into an adult. Number 14. A tilapia strain developed by combining selection and DNA genotyping technology. A. Gift or genetically improved farmed strain. B. Fast or FAC selected tilapia strain. C. Get Excel or genetically enhanced tilapia with excellent qualities strain. D. Genomar strain. Answer: C. Get Excel strain. Get Excel tilapia is a new breed developed by the government, BFAR, by combining improved breed of Oreochromes niloticus using within family selection and rotational mating scheme. Number 15. A technique of determining genetic diversity between and among individual or population by way of separating particles. The example of this is proteins, with different electrical charge. A. Electrophoresis. B. Gene mapping. C. Cloning. D. Gene marking. Answer: A. Electrophoresis. Electrophoresis is a laboratory technique used to separate DNA, RNA, or protein molecules based on their size and electrical charge. Number 16. The mixing of genes between species or strain. A. Genetic introgression. B. Genetic drift. C. Mutation. D. Gene flow. Answer: D. Gene flow. Gene flow, or gene migration, is the transfer of genes from one population to another due to the movement of gametes, individuals, or groups of individuals. For example, people that move to new cities or countries, or pollen that's blown to new flowers. Number 17. A cross of bighead carp and silver carp is an example of A. Interspecific hybridization. B. Intergeneric hybridization. C. Hybridization. D. Crossbreeding. Answer: D. Crossbreeding. Crossbreeding involves the mating of animals from two breeds. Normally, breeds are chosen that have complementary traits that will enhance the offspring's economic value. Number 18. Crossing of individuals from different species. A. Hybridization. B. Interspecific hybridization. C. Intraspecific hybridization. D. Outcrossing. Answer: A. Hybridization. Hybridization is the mating of genetically differentiated individuals or groups and may involve crossing individuals within a species, also known as line crossing or strain crossing, or crossing individuals between separate species. Number 19. A sterile fish. A. Tetraploids. B. Triploids. C. Diploids. D. Haploids. Answer: B. Triploid. Sterile fish are also a prominent tool for biological control of non-native fish. The most common and practical methods currently used to induce sterility in the aquaculture industry are chromosome manipulations to cause triploidization. Number 20. The observable features of an individual organism that results from an interaction between genotype and environment. A. Morphology. B. Phenotype. C. Body color. D. Growth. Answer: B. Phenotype. Phenotype: all the observable characteristics of an organism that result from the interaction of its genotype or total genetic inheritance with the environment.